This is going to be Chapter 20, Part 1, Development and Inheritance. An overview of topics in development. Development, the gradual modification of anatomical structures and physiological characteristics during the period of conception to maturity. Development involves two things: the division and the differentiation of cells. When the cells differentiate, essentially they come from the same stem cells. As these stem cells develop and they differentiate, they become more specialized. The heart, the lungs, the liver, intestines, connective tissue, and so on. And the changes that produce and modify anatomical structures. Differentiation, the creation and of different types of cells. Development begins at fertilization or conception. Embryological development involves the events that occur in the first 2 months after fertilization. And then we have fetal development, another term, and this begins at the start of the 9th week and continues until birth. Fertilization, we're going to talk about activation of an oocyte and the overview of prenatal development. Fertilization involves the fusion of two haploid gametes, which again, if we'll remember from the last lecture, half. So this is half from mom and half from dad, which produces a zygote, and this is the one cell that everything is going to derive from, that contains 46 chromosomes, and that's the normal number of chromosomes for a somatic cell. The activation of an oocyte. The acrosomal cap of the sperm contains several enzymes, including hyaluronidase, hyaluronidase. Dozens of sperm cells must release hyaluronidase before it opens the opening forms between the follicle cells. And this is essentially it's a secretion that occurs on the outside of the oocyte, and it'll allow for the sperm to enter inside and fertilize the cell. Upon contact with the membranes, the sperm cell and the oocyte begin to fuse, triggering oocyte activation. The male pronucleus and the female pronucleus then fuse in a process called amixis. And this is an overview. So this is the oocyte at ovulation. Zone of pellucidea, right here. This is from the previous chapter. The oocyte is suspended in metaphase of meiosis II. You can see the spindle on this cell. Acrosomal enzymes and multiple sperm create gaps in the coronal radii. And you see how they're kind of working on this until they can get in. Once we get one of them that's in there, it'll start the activation process. A single sperm then makes a contact with the oocyte membrane, and the membrane fusion occurs, triggering oocyte activation and completion of meiosis. The sperm is absorbed into the cytoplasm, and the female pronucleus develops. Be this. Number 3, male pronucleus develops, and the spindle fibers appear, and that's here, and these are fixing to divide in preparation for the first cleavage division. Metaphase at the first cleavage division. These are all the chromosomes lined up, and this is 2 haploid cells at this point, half and half from mom, half from dad. And then the blastomere develops. Now these things here, as we see development occurring, this is going to divide again and again and again until we get a multicellular object. Overview of prenatal development. Gestation. Time spent in prenatal development, and we term this in trimesters. First trimester, period of embryologic and early fetal development. Basic components of each of the major organ systems appear. So they may not be functional yet, but they start to appear and they start getting in line. So the cells begin to differentiate. Second trimester, dominated by the development of the organ systems to near completion. Fetus body proportions change. By the end of the second trimester, it looks distinctively human. Third trimester, Characterized by rapid fetal growth. Early in the third trimester, most of the major organ systems become fully functional, and the infant born 1 to 2 months premature has a reasonable chance of survival. So at the end of this, we'll kind of overview an entire timeline and kind of take a look at it. There's periods in here to where we can develop pretty major congenital malformations. We worry about things that would affect fetal development, and we'll talk about that a little bit too. So the first trimester. First trimester is the most dangerous period of prenatal life. Only about 40% of conceptions produce embryos that survive this period. Cleavage and blastocyst development. Cleavage, which is the separation into two inner areas before the cell separates and becomes multicellular, is a cell division that begins immediately after fertilization. The blastomeres, the first cleavage division, produces 2 daughter cells. This is completed about 30 hours after fertilization occurs. And this is the first cleavage here. Polar bodies, you still have them here. Day 1, day 0, fertilization. If we'll note down here on this, fertilization occurs right after the fimbriae. Now here's the problem. Fertilization occurred here, so this developing egg, zygote at this point, as it travels down the fallopian tube, our hopes is that it does not implant like this in here, or this is going to be an ectopic pregnancy. There's only so much real estate on the inside of that fallopian tube. This is where it needs to develop because it has room. Second stage, we have 2 daughter cells. These divide again, these divide again. Daughter cells, blastomeres, dividing, until we get an advanced moural, which is essentially a multicellular wad of cells, and then these start to become more differentiated. So some of them know what they need to do. This is going to adhere to the uterine wall. This is where we're going to start to get our development of the placenta. The rest of this, at the end of this, will start to developing the fetus. It'll grow into here. This eventually will become the amniotic sac area as real estate goes away, and this will become the area that adheres to the side of the placenta. Clinical note, ectopic pregnancy. Fertilized egg normally is implanted in the endometrial lining or the uterine wall. 95% of ectopic pregnancies are implanted in the fallopian tubes. Predisposing factors of this is if they've had pelvic inflammatory disease or an intrauterine device. This may complicate matters or actually precipitate implantation in the fallopian tube. Implantation begins at the surface of the blastocyst, closest to the inner cell mass, touches and adheres to the uterine lining. And we're going to talk about this. I've got some pictures here in a minute. This is figure 23 in the book, and this is the endometrial zone. This is the inside of the uterus, and this is the multicell organism, or the blastocyte. Now, as we showed you earlier on that picture, whenever that cell, that inner cell mass, starts to differentiate, that's what's actually going to adhere to the uterine wall. And as this develops, this will develop in 2 forms, in 2 ways. One, it'll develop in that direction. Chorionic villi will develop, trophoblasts, these cells start to differentiate into the support structure, and this part of it develops into a fetus. This is day 6, 7, 8, and day 9. Formation of the amniotic sac. Trophoblast separates gradually and enlarges, which creates a fluid-filled chamber called an amniotic sac. So this is where, from this point on, we're going to get the 2-way development. One towards the endometrial lining and the other one towards the actual fetus or the developing embryo. Gastrulation and germ layer formation. Gastrulation, some of the cells of the inner cell mass exposed to the amniotic cavity move towards the center line. This movement creates 3 distinct layers called the primary germ layers. Primary germ layers are the ectoderm, the endoderm, and the mesoderm. The ectoderm, superficial layer in contact with the amniotic cavity. The endoderm, the layer facing the blastocele, mesoderm, poorly organized layer of migrating cells, and this is the one that's going to adhere and attach. This is essentially inside out. Alright, so this is a cross section here. We'll come to this here in a second. So this is the same blastocele that came down and adhered to the endometrial lining. Those inner mast cells. This right here develops into a yolk sac. So this is where I was talking about that we have development running in two different directions. Cellular trophoblast, blastocele here, lacunae, cellular development, and this is a cross section of it. So this is the endoderm inside, the mesoderm, which is essentially the middle, and the ectoderm. That was figure 20.4 in your book. The formation of extra embryonic membranes. We have the yolk sac, the amnion, the allantos, and the chorion. Yolk sac, the first embryonic membrane to appear, becomes the important site for blood cell formation. Amnion. The amnion encloses the amniotic fluid and surrounds and cushions the developing embryo and fetus, and we know that. This is going to be the amniotic sac. The allantos, sac of the endoderm and mesoderm, later gives rise to the urinary bladder, and this is the developing fetus. The chorion. The chorion creates a rapid transit system that links the embryo with the trophoblast, and this is where chorionic villi, and these are little villi. When in the future the placenta is going to the only vascular hardwire that occurs between mom and developing fetus or little baby is going to be these little chorionic villi. So all of this actually occurs through diffusion onto the endometrial lining. There's no there's not a vessel that comes in, goes into the placenta, and then to the baby. None of that. All of this occurs, nutrients, oxygenation, through diffusion, and it occurs through the placenta. The placenta is a support structure for the baby. Placentation. Placenta, temporary structure in the uterine wall that provides a site for diffusion between the fetal and maternal circulatory systems. As spelled, my apologies. Placentation, placenta forms taking place where the blood vessels form in the chorion around the periphery of the blastocyte. This is going to be on the outside sections. We'll see a picture of it here in just a second. Umbilical cord, the fetus is connected to the placenta by an elongated umbilical cord which contains the allantos, placental blood vessels, and yolk stalk, and this is going to be support structure for the developing fetus. Second week, we have amnion, which will be amniotic sac, cellular trophoblast, developing area here. This is going to be the placenta at the point that it adheres better. Week 3, let's just kind of follow it and we'll see. Amniotic sac starts to develop more. This is where kiddo is going to be at. Lanteos is in the midsection here. Head of the fold of the embryo. All of this stuff is adhered, but it still isn't giving us tons and tons of feedback structure at this point. And I'll kind of explain that. Chorionic villi of the placenta. These are little finger-like projections that now allow for surface area. Now, 5 weeks from the previous chapter, we also noticed that whenever the luteal phase occurs, the corpus luteum sticks around for about 5 weeks if the egg is fertilized. Now, what the corpus luteum does is it secretes lots of progesterone. The progesterone helps the endometrial wall lining become more vascular. So week 4, embryo now has a head fold and tail fold. Construction of the connective tissue between the embryo and surrounding trophoblast narrows the yolk stalk and stalk body. So this is in development. This is in development at this point. So we got kind of a two-way development occurring here. This is week 5. So at this point, we've provided structures. We have an umbilical stalk. This is going to be a structural supply line for developing fetus. The placenta is developed. We have a yolk sac, chorionic villi of the placenta. Now all of these are now really starting to get involved in this. Chorionic villi. In week 10, we have a definite placenta that now is supportive for the developing fetus. This is where it adheres to the actual uterine wall. This is also the supply line. We have the amniotic sac, which gives a support structure for the developing fetus. And we still have the chorion here, which this is going to differentiate to the amniotic sac. And we'll get, by the time that towards the end of 40 weeks, we'll only have placental adherence here, and then a thin membrane here. All of the nutrients and sectional layers have went to the developing fetus. Placentation continued. Placental circulation. Deoxygenated blood flows from the developing embryo or fetus to the placenta through a paired umbilical arteries. Now, just so that no one gets confused on this, the deoxygenated blood runs through the umbilical artery, and the oxygenated blood return to the embryo in a single umbilical vein. So this is flipped. Deoxygenated blood is running through the arteries, and oxygenated blood is running through the veins. Placental hormones. Human chorionic gonadotropin appear soon after the implantation, indication of pregnancy, which is an HCG test. Most normally, these are the same ones that you can get at the drugstore. They look for HCG in the urine. Human placental lactogen help prepare the mammary glands. Relaxin is secreted by the placenta, and the corpus luteum help the pelvis to expand during delivery. Now, all of these are going to give chemical signaling to expand, continue development. Let's be sure that you have ample blood supply. All of these are giving the same signal to the uterus area. Figure 20.2, and this is early development, but this is actually a blow-up of how the chorionic villi work. Now, adherence occurs in the uterine wall. If you look at this one back behind here, thickening the uterine wall, which is what the corpus luteum does, and then this provides diffusion for oxygen. Works the same as our lungs essentially do, to where it goes from an area of higher concentration to an area of lower concentration. And same way, the returning supply, which in this case is in the arterial area, higher concentration of expelled waste, lower concentration of expelled wastes. Figure 27, and this is 2 weeks into it, as we see the development. This is 4 weeks, starting to look like something at this point. 8 weeks, can kind of make out that it's humanoid. And by 12 weeks, we definitely can see that this is going to be a little bambino. These are very small. This probably isn't no bigger than the size of a quarter, maybe a little larger at this point. Embryogenesis. Embryogenesis is the process of embryo formation. Organogenesis, first trimester process of organ formation. So all of the organs are differentiating cellularly from the environment, and they are becoming more specialized. Clinical note, teratogenicity. Most vulnerable to birth defects in the first 60 days. First stage of gestation, conception to 18 days is the most crucial. Any chemical disease that can cause malformation of the fetus. During organogenesis, or whenever these are actually the organs are performing, the fetus is not susceptible. So at this point, before that, the fetus is essentially compiling all the nutrients and things like that from the placental area that it's going to need to develop. Now, during this time, it's pretty vulnerable. But after it's got everything and it goes through the cellular divisions to differentiate, it becomes less susceptible. Now, there are still medications out there that can run across this barrier. That whole barrier that we looked at about 3 slides ago, and I'm going to back up here just a second. This barrier here, the placental area, detoxification would occur from our body, since this is going to interact with the liver as an organ would, portal circulation system. Now, with that said, during the first start of development, whenever it's getting all the supplies, if a toxic drug got into play at that point, then it would adjust the development of the organs. After that point, whenever it's kind of not really paying attention to what's on this side except for a list of nutrients, and it's developing more from this direction, it's less susceptible. However, there are medications that could cross across this barrier still and cause major congenital malformations. All right, the second and third trimester. The effects of pregnancy on the maternal system. Clinical note on this, physiology changes in pregnancy, structural and functional changes in the uterus, and then fetal development. During the third trimester, the basic components of all organ systems appear. And this is during the third trimester, this is going to be functional. These organs are going to start to functioning with what they actually are supposed to do. Most become ready to perform their functions. The rate of growth starts to decrease, however. The largest weight gain occurs in this trimester. So first, second, I know what I want to do with my life, and then in the third trimester, we get developing growth and the finishing off of the organ systems. The effects on pregnancy on maternal systems. Generally speaking, and most of this will make sense if you think about the demands put up on the body, you have another organ that has grown inside the uterus, and from that, it's going to need resource. So respirations go up, oxygenation, blood volume increases. The fluid that has been added kind of causes a dilution. So this is the anemias that we see whenever we're talking about that a mother that is expecting, or that a female that is pregnant, they can become anemic. And the reason they can become anemic is because of the fluid that has been added essentially dilutes their blood. Remember that the red blood cells are formed elements. So by adding more fluid to the base, we thin out the blood supply. Maternal requirements for nutrients and vitamins climb about 10 to 30%. Glomerular filtration rate increases by roughly 50%. The uterus undergoes an increase in size, and the mammary glands increase in size and secretory activity. So they're going to become prepared to deliver colostrum or mother's milk to the baby. Figure 29. 4 weeks into it, we were nothing more than a little pack of cells. 8 weeks into it, we can differentiate. You can see the fontanelles there. Hands and feet have developed. 16 weeks into it, organ systems are well on their way. And then this is the growth spurt that occurs from a newborn to an adult, which the growth pattern, considering that we came from one cell, is immense. It's absolutely amazing whenever you think about the scope of this. Clinical note, physiological changes during pregnancy. Now, obviously, the biggest physiological change that would occur is to the reproductive system. Mammary glands get involved, tons and tons of endocrine hormones. Uterine wall thickens, becomes more of a supportive structure, starts to grow. The fundus itself thins out and it has the ability to stretch. So as the fetus develops in the uterine area, it starts pushing up and displacing other organs. Newborn expecting mothers at the point that they're 40 weeks into this may even be short of breath because of the displacement of the rest of the organs up against the diaphragm, not allowing for full expansion of the lungs. The pressure on the bladder makes the urinary sensation increase because of pressure receptors on the actual bladder wall itself. Cardiovascular respiratory system decreases airway resistance 20%, increasing consumption, and 40% increase in tidal volume. So we increase, but yet we're decreasing the area that the lungs have to actually expand. Cardiovascular system output increases, blood volume increases by 45%, rate increases by 10 to 15 breaths per minute, blood pressure decreases in the first 2 trimesters, almost normal during the latter of the third, or whenever kiddo is being born. Gastrointestinal system, nausea and vomiting from hormones. Peristalsis slowed. This leads to constipation and heartburn, which are pretty common, and the organs are generally compressed due to the development of the fetus. Urinary system, renal blood flow increases, glomerular filtration rate increases, urinary frequency is common from displacement. Musculoskeletal system, loosening of the pelvic joints, gravity shifts causing lower back pain. And this is 3 months, as we can see at 8 months, and this is a month before delivery. That fetus has developed, and it is essentially went in multiple directions. That way, and that way. And all of the organs now are displaced up. Since the diaphragm is right here, this is decreasing the area for the lungs to expand. Hopefully kiddo is down at 9 months. As the kiddo starts to grow and the last portion, hormones get released, this causes more thinning and dilation of the actual cervix. There is a mucus plug that is in the end of this. As this reaches 10 centimeters, this will thin out in a face. By then, generally the mucus plug will have either pushed through or the water has broken. That will bring about more pitocin, which will cause contraction of the uterus to expel kiddo. Figure 2011 in your book. Blood volume usually increases by about 45%. Dilution resulting from the disproportionate increase in plasma volume over the red cell mass is responsible for the so-called anemia of pregnancy. Cardiac output increases by 1 to 1.5 liters per minute during the first trimester, reaches 6 to 7 liters per minute by the late second trimester, and is maintained essentially at this level until delivery. Stroke volume progressively declines to term following a rise early in pregnancy. Heart rate, however, increases by an average of 10 to 15 beats per minute. So, stroke volume declines, heart rate increases, cardiac output increases, and then our actual blood volume goes up, but due to the plasma dilution, the female appears anemic. Structure and functional changes in the uterus, a rise in estrogen, a rise in oxytocin, and then prostaglandin production. Now, this is figure 2012 in your book. So, we're going to start on this end and work our way back and kind of explain this. So, at the point that the development feels that we're ready to give birth, or the kiddo has grown enough and we get a release of oxytocin from the pituitary. This is going to bring about increased prostaglandin production itself. It will cause an excited area or an excitability of the uterine musculature, and this would be the actual uterus itself. And then when the contractions start occurring, this is a positive feedback system. Now, The positive feedback system on this essentially reinforces the initial thought. So once this begins, the only way to stop the process is to remove the fetus, very simply. Labor contractions in the positive feedback system will trigger this distortion of the myometrium, which now starts to expel things, increase prostaglandin production. So we're going to be in this loop once it starts from the release of oxytocin. One of the things that we see if a person is having a hard time with the contractions is we will give them an artificial version of Pitocin or synthetic version of oxytocin. As fetal growth occurs, this also gives a feedback system to the myometrium, which increases prostaglandin production and increases the excitability of uterine musculature. This is why we get Braxton Hicks sometimes. So as it's developing, the muscles may spasm a little, which are false labor pains. Placenta production of estrogen and relaxin. The relaxin actually allows for the expansion of the myometrium, or the muscular layer, or the thick layer, the internal layer or inside layer of the uterus. Estrogen, maternal oxytocin released at the posterior pituitary is what gives that feedback, which again reinforces this idea. So all of this, once we get started and it gets development, is hormonal, and the hormones themselves set up the environment so that the actual uterine wall grows and expands appropriately at the point that the delivery starts, or that we are at 40 weeks, hopefully, or near 40 weeks. It is reinforced again and again and again through the positive feedback system. Example, first contraction hits, myometrium is irritated, releases more prostaglandins, increases the excitability, placenta is squeezed a little bit, which releases maternal oxytocin, increased excitability. Now the next contraction occurs and they become progressively stronger. So first contraction, second contraction. Now all of these are going to reinforce the idea until the fetus is expelled from the uterus. As far as fetal development goes, the red areas are the areas that we are concerned with major congenital abnormalities. So in the central nervous system up to the 16th week, at the end of the 16th week, we can have major congenital abnormalities. Now after that, you can get minor ones and functional defects. So from 20 weeks or so, and this is where we worry about the tetranogicity of medications, that they can cross the placental area and affect the developing fetus. Heart, about midway through 6 weeks. Upper limbs, just before the end of the 5th week. Eyes, halfway through 8 weeks. Lower limbs, just after 5 weeks. Teeth, end of 8 weeks. Palate, just after 8 weeks. External genitalia, just before the end of 9 weeks. And the ears, halfway through the 9-week area. So very simply, the red areas are where we can, if we had a drug that had tetragens in them and it was implemented during the 4th week, we would be concerned in the fetal development. This ends part 1. If you have any questions on it, feel free to contact me. My name is Roy Smith, 405-219-7613, or smith@imsa.net. Thank you.